Hello again. Okay, so today we're going to continue with our urinary system lectures, and we're going to focus this lecture on the regulation of GFR. So what is GFR? What does it stand for? So glomerular filtration rate. So we're talking about how quickly filtration occurs inside the kidney, inside the nephrons, right? Okay. So glomerular filtration rate is that volume of filtrate that's formed per minute by the kidneys. So if you have watched the first couple of lectures, you know that you have blood flow that comes in an afferent arteriole, correct? It goes into this glomerulus, which is a capillary bed. Around that capillary bed, you have these podocytes, which kind of form the filtration membrane, kind of the sieve. And so as blood flows in, due to the forces of hydrostatic pressure pushing out and osmotic pressure kind of sucking back in, you get the formation of filtrate, which is basically plasma minus the proteins and minus the big proteins. And that's going to be processed through steps of reabsorption and secretion through the nephron and finally be in its final form as urine. Okay? And so that is what glomerular filtration rate is how much of this filtrate is being formed at per minute that can eventually be processed and turned into urine. Now remember, all filtrate does not become urine because of that process of reabsorption and secretion. Really only less than 1% of what is filtered out is actually excreted out of the body as urine. We get most of that back. We want to reserve our water and our salts and our sugars and all that kind of thing. We want to hold on to the good stuff and just get rid of the bad stuff, okay? So the way we regulate glomerular filtration rate is really done in two ways. There's two homeostatic mechanisms that use the negative feedback and how these are going to control glomerular filtration rate. And one is an intrinsic control, so intrinsic, which means it's renal autoregulation. It's just happening locally at the level of the glomerulus and in the kidney. Okay, so just locally inside the kidney at the nephron glomerular level, and doesn't require any hormones or anything else. There's no outside forces that are acting on that. The other one are the extrinsic controls, and this is where the nervous system and the endocrine system play a role in maintaining GFR as a means to regulate blood pressure. Okay, that's kind of the main reason that they're going to be involved, is to change how much water your kidneys are getting rid of or holding on to, because we know that the volume can affect blood pressure, right? So the amount of water in your body affects your blood volume, which affects your blood pressure. So this can regulate GFR either at an intrinsic level or an extrinsic level. And we're going to walk through both of these processes today. So your intrinsic controls, this is what's going on all the time. Okay, so this is that day-to-day, minute-to-minute control of your kidneys and their GFR rate. And this happens when your mean arterial pressure is in the range of about 80 to 180 milligrams per mercury. So, you know, normal blood pressure, a little high, 180 is pretty high. But, I mean, you know, that's still in times of even stress. I mean, your blood pressure doesn't get that high. So this is going to be able to regulate your blood pressure during what you would consider your kind of your normal range of blood pressure, even though it does seem a little high. But that's good. You have a really wide range of pressure that your kidney can control this without having to rely on the nervous system and the endocrine system. It can kind of take care of itself. And it works in two ways. You have two types of renal autoregulation, or two types of these intrinsic controls. You have a myogenic mechanism and a tubuloglomerular feedback mechanism. So myogenic. So think back to 201, your first anatomy, 201. Or wherever you had anatomy, whatever your first anatomy was. Myo, what does that tell you? What are you thinking? What does myo stand for? Muscle. So this is something to do with some kind of muscle. Now, I don't necessarily mean cardiac muscle or skeletal muscle. I'm thinking more along the lines of smooth muscle, okay? So this is something, so smooth muscle is found where? Inside of tubes. So automatically you're thinking, oh, there's blood vessels, those are tubes, they have smooth muscle. So this is some kind of contraction and vasodilation of the smooth muscle in these small arterioles associated with the kidney, Okay, so just looking at the word should go, oh, okay, I know what that means. The tubuloglomerular feedback mechanism means that somehow the tubules and the glomerulus are in contact with each other. Somehow they're communicating and making changes. Okay, and this is done at that place called the juxtaglomerular apparatus. That's why I call it the JGA, and everybody else did too, because it's hard to say. So at the JGA, the glomerulus and the tubules are in contact with each other, and there can be a feedback regulation mechanism there. Okay, so let's talk about that myogenic mechanism first. So in response to high blood pressure, let's say your blood pressure has risen over a short time, and so what would you do? Blood pressure is high, so that's going to be forcing blood through the kidneys, right? Well, the glomeruli, these little capillary beds where filtration occur, are pretty delicate, and so you want to protect those. And so if blood pressure is high, what that actually causes is a constriction of the afferent arterioles. So let me go to a picture. So remember, the afferent arterioles is the way blood goes into the glomerulus, right? Well, if it's coming in at too high pressure, you could blow out the glomerulus. So you want to reduce that pressure by kind of shutting off that blood flow and reducing the flow and whoops, which way? There. And basically kind of reduce that amount of GFR, kind of decrease that flow rate and decrease the pressure at the glomerulus. So that helps maintain normal GFR without damaging the glomeruli from that high blood pressure. Now, this is one of the things that over time, eventually this myogenic mechanism just can't keep up. So people that would have chronic high blood pressure and that's not treated, even if it is treated and it just stays high and they just can't seem to get it down, constant high blood pressure finally puts pressure on those glomeruli and they start to blow out and scar. So that's one of the reasons people that have long-term chronic blood pressure wind up on dialysis and with kidney problems, because that high blood pressure is just wreaking havoc on their glomeruli, and so the kidneys just can no longer function correctly. So that's one of the side effects of high blood pressure. It's also a side effect of diabetes. Diabetes is notorious for damaging small capillaries. It's one of the reasons that people go blind when they have long-term diabetes, and one of the reasons they have kidney failure is because those capillaries are damaged over time as a side effect of diabetes. Now, so all of that was kind of an aside, but we're talking medicine, so might as well start bringing things together in as many organ systems as we can. That's the whole point of this class, is to start assimilating everything we've learned in the different organ systems and trying to make it tie all together. You just had endocrine, so you should know something about diabetes, right? And so that's high blood sugar. And again, one of the results of diabetes is leaky capillaries and damage, especially to the eyes and the kidneys. Okay, sidebar. What happens if you have low blood pressure? So if you have a low blood pressure is falling, we want to dilate those afferent arterioles, increase the blood flow so that more blood's going into the glomerulus and able to maintain that filtration rate and keep GFR normal. So that's the myogenic control. So increased blood pressure is going to constrict the afferent arteriole and reduce blood flow to the capillary in order to reduce GFR, help maintain it so it doesn't get too high. Low blood pressure is going to dilate and open up the afferent arterioles to allow more blood flow and to keep the filtration rate going. Okay? It's a myogenic mechanism. So that's just happening right there at the kidney. You don't need any nervous system or endocrine system to do anything about that. Now the other type of intrinsic control is your tubuloglomerular feedback. So tubuloglomerular, so we're knowing the tubules and the glomerulus are somehow talking to each other. And that happens at the JGA, which is the juxtaglomerular apparatus. So if you remember back to when I was talking about the kind of the overall anatomy of the kidney, here's your glomerular capsule, here's your glomerular capillary, your glomerulus. You have your afferent arteriole, your efferent arteriole where blood is going out. This is going to be the proximal convoluted tubule, and if you remember, the proximal convoluted tubule does that wavy stuff. It makes a long loop of Henle, then it makes another wavy thing called a distal convoluted tubule, and then that's going to go into a collecting duct, and then that's going to go out to the ureters. Okay. So where the ascending limb of the loop of Henle and the distal convoluted tubule, that transition area, that actually comes back to its own glomerulus. So this right here, this is that transition area between the ascending limb of the loop of Henle and the distal convoluted tubule. Now I have to say something because this is kind of one of those things that drives me nuts. It really depends on which book you're reading. In some books they say that the JGA is in the distal convoluted tubule. Some say it's at the portion of the thick ascending limb. It's really at that transition point. So I learned it as part of the distal convoluted tubule. Some textbooks call it the thick ascending limb. It's really at that junction point. So it's kind of, they're one and the same. So don't get too caught up in, if you read in one book it says it's thick ascending limb and one says it's distal convoluted tubule. It's right at the transition point. But what's really special about that particular area of the tubule, where these, what's called the juxtaglomerular apparatus, is in this portion of the tubule, you have some specialized cells called macula densa cells. And so they're going to be right here. They face the lumen of the tubule, and they also are in contact with something called granular cells. And mesangial cells. So you have mesangial cells here, and then these granule cells right here. So there's really kind of three cells all together that make up this juxtaglomerular apparatus, or JGA. Macula densa cells, mesangial cells, and granular cells. Okay, and we're going to talk about what all those do. There's actually another set of mesangial cells in the kidney I want to talk to you just briefly about. They're the mesangial cells that are between the capillaries, which are these purple things. And mesangial cells are muscle cell-like. They're able to be a little contractile, so they are able to help in opening and closing off the glomerulus. So they can contract and relax, kind of change the shape of the glomerulus if you're talking about these mesangial cells. So there really are two different mesangial cells, those that are found in the JGA and those are found in the glomerulus, just in case you're reading that in the textbook and see that. They're two different sets of mesangial cells. Full disclosure, I spent 6 years of my life studying these, and so they're there. They're a real thing, but we won't talk a whole lot about what they do. You don't want to hear 6 years of my life, do you? I didn't think so. All right, so the JGA. So what's going on? You have one per nephron, okay? And what its job is to regulate the filtration formation and blood pressure. So it's regulating how quickly filtrate's being formed, which in turn will regulate blood pressure. All right, so if you're making a lot of filtrate and you're going to get rid of a lot of filtrate, you're going to lower blood volume. What's that going to do? Lower blood pressure. If you're not making a lot of filtrate, you're not making a lot of urine, what are you going to do? You're holding on to water, you're going to raise blood pressure. So these are in tune with blood pressure regulation. The JGA, again, involves that portion of the tubule. Again, in your book, it says the ascending limb of the loop of Henle. It's also the beginning of the distal convoluted tubule. I don't get too caught up in the specifics. It's right there at that transition point. And the afferent arteriole. You have granular cells, which are also called juxtaglomerular or JG cells. They're the same thing, so you may see either one of those depending on where you're reading. These are the cells that have an endocrine function, really. I mean, they secrete something called renin. And so we'll talk about what renin does in the idea of angiotensin and that kind of thing. So we'll talk a little bit about that later. We talked about the endocrine system earlier, about how you have the kidneys making EPO and renin. So the glomerular cells is where that renin is made. These granular cells also act as like mechanoreceptors. So think of a baroreceptor in the aorta. So it's going to sense blood pressure. Then you have your macula densa cells. So again, you're looking at your picture. Macula densa cells are here. The JG cells are here. They're going to be tall, closely packed cells that face the lumen of that portion of the tubule, either if you want to call it ascending limb or distal convoluted tubule. They're there, and they have chemoreceptors. So you have something that measures pressure and something that's measuring the chemistry, okay, of the blood, or of the filtrate, And so they are actually measuring how much sodium chloride or how much salt is in the filtrate. So you have something measuring the pressure inside of the area, and you have something measuring the sodium chloride content. And then again, you have these extraglomerular mesangial cells, and it's thought that these cells, because of where they're located, okay, so here, I'm sorry, I did that wrong. These are the granular cells right here. Let me see if I can erase. Hang on, I got too many, whoops, got too many things going. Let's see, whoops. Oh no. Eraser. There we go. I know y'all hate it when I stop and just start messing with the technology. So again, just to be clear, here's your macula densa cells here, okay? Here's your granular cells here, and then here are your mesangial cells. So you can see they're between the macula densa and the granular cells, so they may act in some kind of communication. They may be able to pass signals between the macula densa and the granular cells. So you have some communication between something that can regulate blood pressure, but that's acting by sensing the pressure, and something that's sensing the salt content. So we know that, blood pressure and volume and water and salt, those are all related. So it's a really good thing that these things are talking to each other, okay? And it's happening right here at the JGA. All right, so what's happening? What is the tubuloglomerular feedback mechanism? It depends on the flow. It's a flow-dependent mechanism. So basically the flow of the filtrate that is directed by the macula densa cells. So what do the macula densa cells do? They are sensing what? Sodium chloride. So as filtrate is passing by the macula densa cells, they're measuring how much sodium chloride is in the filtrate, and in response to that level of sodium chloride, they're going to change the GFR. They're going to either increase filtration rate or decrease filtration rate. They're going to send a signal, hey, we need to get less sodium chloride, we need to reabsorb more sodium chloride, or we need to get rid of more sodium chloride. So they're going to change filtration rate in order to adjust the amount of sodium chloride that they're sensing at that level. Because remember, if you go back and you look at your nephron, okay, so let's draw the little nephron again. Here's your lip of Henle and distal convoluted tubule, and then going down to the collecting duct. You really have one more chance as this filtrate is passing through the distal convoluted tubule and then out to the collecting duct. You still have a chance, right? Based on aldosterone, ADH, and all those hormones, if you need to change how much water is being released. You can either resorb more water or get rid of more water. You have one more chance. So by kind of measuring the amount of sodium chloride here, it kind of gives you a chance to know what you got to do. We can either increase the filtration rate, decrease the filtration rate, and then possibly the endocrine and nervous system, the endocrine system can jump in and help us with holding on or releasing more water. So this is a pretty complicated process, and I'm going to show you an overall picture here in just a minute. I kind of got ahead of myself. So what is the tubuloglomerular feedback? So okay, it's the macula densa cells measuring the flow rate and the sodium chloride concentration in the filtrate. So if your GFR is increasing, so it's kind of going fast, that means the filtrate is flowing pretty quickly in the tubule. And it's flowing so high, it's going so fast, there's really no time for sodium chloride reabsorption. So it's kind of flowing through the tubule so quickly you don't have enough time to reabsorb the sodium chloride out of the filtrate. That means the sodium chloride concentration is going to be high. There's going to be a lot of sodium chloride because you just didn't give it enough time to be reabsorbed. So those macula densa cells are going to respond to that high sodium chloride, and they're going to release a vasoconstricting chemical that acts on the afferent arteriole. What happens if you constrict the afferent arteriole? If you constrict this, what happens? You reduce flow rate, right? You decrease GFR. So if the macula densa cells sense too much sodium chloride in the tubule, they will send a chemical to the afferent arteriole to constrict, cut off kind of the blood supply to the glomerulus, slow it down, and decrease the GFR. That's going to slow the flow rate through the tubule, giving time for sodium chloride to be reabsorbed. Okay? just the opposite if GFR decreases. You just do the opposite thing. You speed up GFR. So if sodium chloride is too low, then that means it's going through too slowly. You're reabsorbing too much sodium chloride. You want to speed it up, okay, so that you can get rid of more sodium chloride. So those are the two intrinsic, so the myogenic and tubuloglomerular feedback. So what happens when you need the nervous system and the endocrine system to play a role? Well, that's your extrinsic controls. And so these are also at play during normal conditions at rest. So in normal conditions, when you're at rest, the normal nervous system endocrine role is really to just to do nothing, I guess you would say. They're just kind of staying at homeostatic levels. And so you're kind of in parasympathetic mode. And so your renal blood vessels are dilated, and you don't have to send out any extra hormones or any extra nervous system signals to do anything. They're just kind of at their I guess, normal levels. And so what you're doing is letting your intrinsic regulation kind of do all the work. But under times of stress, when your sympathetic nervous system needs to kick in, okay, so remember, exercise is stress, alien walking in the door is stress. Whenever you're stressed, you're going to get that sympathetic kick, and that's going to include, the epinephrine, norepinephrine stuff. So you have norepinephrine is going to be coming from the sympathetic nervous system, epinephrine from your adrenal gland, so this is kind of nervous system and hormonal, from the adrenal medulla. So you're trying to increase blood pressure typically in a sympathetic event, right? Increase blood pressure so you can increase blood flow out to the heart and muscles so you can get away from the alien or exercise or whatever you need to do. However, in the kidney, both of these cause constriction of the afferent arterioles, okay? Because we're really not too worried about the kidneys functioning right now. We don't want to get rid of all of our water. We want to hold on to blood volume to keep blood pressure high. So we want to basically inhibit filtration or decrease filtration. So we decrease how much water we're getting rid of. So we hold on to that. That way we can keep blood volume high and blood pressure high. So it's going to inhibit filtration and cause the release of renin. So what's renin? So renin is an enzyme that will convert angiotensinogen, which is found in the blood, to angiotensin 1. And there's another enzyme called angiotensin converting enzyme, called ACE for short, that converts it to angiotensin 2. And this is the active thing that we're going to talk about. Okay? So anytime you need to raise blood pressure, so in a sympathetic event, or if your blood pressure falls too low, if your mean arterial pressure falls below 80, which remember I gave you that range of 80 to 180. If it falls below 80, then something's happening. Something's causing your blood pressure to fall. Well, you're going to pass out. You're going to have to get your blood pressure back up. So a triggering event of a low blood pressure below 80 will also trigger the JGA cells or the JG cells to release renin. So renin is going to be something that will increase blood pressure. And it does that by a cascade effect, which results from the production of angiotensin 2, which a lot of times you'll see written as just A2. So what does angiotensin 2 do? You may have seen this earlier in some other blood pressure talks, but what it does is it will constrict smooth muscle, and we're talking about all your vasculature, your peripheral vasculature, where you want to increase blood pressure. You'll start to increase constriction on those blood vessels, and when you constrict these blood vessels, you make the lumen smaller. That causes the blood pressure to go up because it increases peripheral resistance. You remember that? Increase peripheral resistance, increase blood pressure. Okay. It also stimulates the reabsorption of sodium. Well, if you're reabsorbing sodium, what's following sodium? Water. If you're holding on to water, what's going to happen? You're going to increase blood volume. What's going to happen if you increase blood volume? You're going to increase blood pressure. So increase peripheral resistance, increase blood volume, increase blood pressure. It causes the adrenal cortex to release aldosterone. Aldosterone has something to do with sodium. Sodium and water. Here we go again. It stimulates the hypothalamus to release ADH. You guys know what that is. Anti-diuretic hormone. So if you're anti-diuresing, you're not peeing, you're holding on to water, you're increasing blood volume. What are you doing? Increasing blood pressure. It causes the efferent arterioles to constrict, which is going to change the capillary hydrostatic pressure and increase fluid reabsorption. Okay, so it's going to work at the level of the glomerulus. It also causes those glomerular mesangial cells, that other set I told you about, the ones that I worked on, they contract, and that lessens the surface area in the glomerulus to decrease the surface area for filtration. So you're not going to filter as much, you're not going to get rid of as much filtrate, you're not going to get as much urine, you're going to hold on to more water, you hold on to more water, you hold on to more blood volume, you do what? You increase blood pressure. So angiotensin II increases mean arterial blood pressure in a bajillion ways, okay? All of these things. That looks like an excellent all-but-apply question, just saying. All right, so let's think of the big picture. How many systems, again, this could be a great essay question. How many systems can you include when you're talking about blood pressure regulation? I know you guys thought, oh, blood pressure, that's a heart thing. It's a kidney thing. It's really both. And so if you're a cardiologist, you think the heart is just the most important blood pressure regulator ever. If you're a kidney doctor, nephrologist, you think that kidney's the most important blood pressure regulator ever. But you know, they kind of work together. It's kind of a team effort. So what can happen if you have low blood pressure? All right, so your systemic blood pressure is low, okay? So low, so like below 80. What do we want to do to get that blood pressure back up? How many ways can we do that? So I love this slide. It shows you your intrinsic mechanisms, and it shows you your extrinsic mechanisms. So everything in blue and gold, or kind of orange, are going to be your intrinsic mechanisms, you know, acting at the level of the kidney. Your extrinsic system is going to include that nervous system and endocrine system. So let's just kind of go through these columns and see how they all relate. So low blood pressure, okay, so remember you have receptors in your body, all right, so you have low blood pressure, that's going to decrease GFR. That decreased GFR is going to decrease the stretch of the smooth muscle cells inside those afferent arterioles. That's going to cause those afferent arterioles to dilate, let more blood go through, let more filtration occur. That increases GFR, okay? So that's that myogenic mechanism. If you have falling GFR, if filtration rate is low, what's going to happen? You're going to have changing sodium chloride levels, okay, in that distal convoluted tubule. Those macula densa cells are going to pick up that falling sodium chloride levels, okay? They're going to cause vasodilation of the afferent arterioles, okay, and allow more GFR, more filtration. Okay, that's the tubuloglomerular feedback mechanism. All right, so that's just happening at the level of the kidney. All right, what happens when you want to pull in the hormones? So low blood pressure, those granular cells, they're going to be stimulated to release renin. Renin's going to do that cascade process that results in angiotensin II. Angiotensin II does several things. It causes the release of aldosterone from the adrenal gland. Aldosterone will go through the blood supply, because it's a hormone, to the kidney tubules. They're going to increase sodium reabsorption. That's going to pull water along. That's going to increase blood volume. That's going to increase blood pressure. Angiotensin II also is going to cause systemic arterioles to vasoconstrict, which is going to increase peripheral resistance, which is going to do what? Increase blood pressure. All right, what about, those are all endocrine, kind of that renin-angiotensin system, hormonal. What about neural controls? You're going to have baroreceptors, all right? They're going to be, they're going to not be stimulated anymore because they don't have a lot of high pressure that's going to be going against them. That's going to turn the sympathetic nervous system on. The sympathetic nervous system will also cause those arterioles to clamp down, increase peripheral resistance, and increase blood pressure. So this is a great slide with kind of everything on it, and shows you all the different systems, the myogenic responses, the tubuloglomerular feedback responses, the hormonal responses, and the neural responses in response to low blood pressure. So you will see some questions that come directly from this slide, because there's a lot of information here. Okay, it means I expect you to know what aldosterone is. I expect you to know what angiotensin is. I need angiotensin II. You need to know what renin is. So if you could take this chart and erase the words and then fill it back in, you'll be golden. That's a lot of information about the control of blood pressure using the kidney. Okay. All right, so that's it. A lot of information. It's kind of a long video, but GFR is kind of a complicated process. So I will see you again. We'll talk more about urine and some medical considerations in some of our future videos.